Okay, so ladies and gentlemen, you are all welcome to my YouTube channel. Today, we are going to look at some questions on post UTME. So the first question said, uh, is the smallest eukaryotes A, virus, B, bacteria, C, plasmodium, D, sponge? So first, we need to understand the meaning of eukaryotes. So generally, we have cell. And what is cell? Cell, we have to define cell itself. So what is cell? Cell is the structural and the functional unit of life. Cell is the structural and the functional unit of life. It means that everything is cell. Everything that is surviving is actually due to cell. And if there is no cell, there is no life. So cell is the fundamental and the functional unit of life. And we have two types of this cell. We have prokaryotic cell and eukaryotic cell. We have prokaryotic, we have prokaryotic cell. That is number one. And number two, we have eukaryotic. We have eukaryotic cells. So these are the two types of cells that we have. Prokaryotic cells are those cells that doesn't have nucleus and membrane-bound organelles. They doesn't have nucleus and membrane-bound organelles. No nucleus, no nucleus. They don't have nucleus and membrane-bound organelles. And examples of these membrane-bound organelles, we have something like mitochondria, Golgi body, ribosomes, and lysosome. So these are the examples of membrane-bound organelles. So eukaryotic cells are those cells that doesn't have nucleus and membrane-bound organelles, while eukaryotic cells are those cells that have nucleus and membrane-bound organelles. So they have nucleus and membrane-bound organelles. So example of prokaryotic cells is bacteria. So bacteria is an example of prokaryotic cells. So therefore, from this option, since we said that bacteria is an example of eukaryotic cell, it means that it is out of this option. And then second, option A is virus. So what is virus? So virus generally is not an organism. That is why we call it particle. So virus is not an organism, it's a particle. It's a particle. Why do you call it particle? We call it particle because virus on it is on, it cannot sustain life. When it is on, it cannot sustain life. So it only depends on the host cells. So it survive in another organisms. So that is the only condition that it can survive in organisms. And these viruses, they are very dangerous. They are very, very small. They are very, very extremely small. And they are not completely living organisms. They are only survive when they are in another organisms. So that is why you have to take it out. So virus is not an organism. It's just a particle. It's just a particle. And in addition, we have plasmodium. So plasmodium is an example of protozoan. Remember that from classification of living organisms, we have three domains. It started from the classification of organisms. We have three domains of classification, and this three domains of classification includes what we call we have eukarya, we have prokarya, and then we have archaea. That is the three domain of classification. The three domain of classification, as I said, we have eukarya, and we belong to this, and we have bacteria, or sometimes we call it eubacteria, and we have archaea. So these are the three domain classification of living organisms. And then this domain further divide into 5 kingdoms. It further divide into 5 kingdoms. Kingdoms. So we have 5 kingdoms, and these 5 kingdoms is based on is based on 7 levels of classification, which is K, K, P, COF, GS. I usually abbreviate it like this: K, P, COF, GS. K stand for kingdom. K stand for kingdom. P stand for phylum. P stand for phylum. C stand for class. And then the O here stand for order. And then the F stand for family. The F stand for family. And then the G stand for genus. The G stand for genus. And S stand for species. And the smallest unit of classification of living organisms, know this, ladies and gentlemen, the smallest unit of classification of living organisms is species. So species is the smallest unit of classification of living organisms. Then we also have genome. And of course, in biology, we have what you call binomial system of living organisms. Binomial system of living organisms, it means that each organism has 2 names. Each organism has 2 names. The first name is generic name. The first name is generic name. We call it generic name. The first name is generic name, which is coming from the genus. And then the second one is species name. So each organism has 2 names. The first one is generic name and the second one is species name. Like for example, human being have a binomial name of Homo sapien. Homo sapien. Homo sapien. So usually this name is either italicized or after you write it, you underline it. Whenever you are writing binomial name or biological classification of living organisms, when you write it, it usually has 2 names. And the first name here is the generic name. That is the genus name. The first name is the genus name. The first name is the genus name, and it's always started with capital letter. And then the second name is the species name. The second is the species, name. The species name, and it usually start with small letter. So each and every living organisms from the classification, it has 2 names. It has 2 names. The first one is generic name, which is started with capital letter, and the second one is species name, which started with small letter. So here we have Homo sapien, Homo sapien, and then we also have Ratus Pantoralio, Plasmodium falciparum. All of them, they are binomial name, or we call them scientific name. They will always have 2 names. So that is in the case of classification. So as I said, we have 5 kingdoms. We have 5 kingdoms. And what are those 5 kingdoms? What are those 5 kingdoms? So we have 5 kingdoms. The first kingdoms is kingdom Monera. The first one is called kingdom Monera, and the second one is kingdom Protista. And then we have kingdom Fungi. We have kingdom Animalia. And then we have the last one is kingdom Plantae. So of course, animals is divided into two. It's divided into two phylum, invertebra and vertebra. And of course, the protista is divided into protozoan, protozoan. So we have protozoan, protista, and then we have photosynthetic protista, photosynthetic. We have photosynthetic protista, and of course, when we said photosynthetic, it means that there are those protista that can synthesize their output. They can synthesize their output, and then that is so. This is one, and then second, and then the third one under this protista we have consumer protista, consumer protista. So these are the three that are fall under protista. We have protozoan protista, we have photosynthetic protista, and we have consumer protista. So these are the three protista that we have. And of course, photosynthetic protista includes dinoflagellates, diatoms, and euglenoids. Euglenoids are under photosynthetic protista. And of course, consumer protista, we have slime molds, we have myzomycetes. They are all under consumer protista. So now, and all this kingdom protista is fall under eukarya. It falls under eukarya. That is eukaryotes. So fungi, kingdom fungi is eukarya. Animalia is also eukarya. Plantae is eukarya. Protista is also eukarya. So of course, another examples of protozoan, another example of this protozoan protista, we have plasmodium. We have plasmodium. So this plasmodium, there are species that are found under this protista. They are actually the well-known species of plasmodium. There are five. There are five, and they are the causative agent of malaria. They are the parasite that cause the disease malaria. They are the parasites that cause the disease malaria. And of course, mosquito, which is what do you call which is so mosquito is the cause, it's a vector that is a carrier that take this parasite from one organism to another. Because whenever we said vector, it means that they are organisms that transfer disease parasites from one organism to another. So of course, one of the species of the mosquito that transfer this malaria is called female Anopheles mosquito. Is female Anopheles mosquitoes. So it is the one that transfer or that carry this plasmodium disease or this plasmodium parasite. So the major species that cause the disease is called Plasmodium falciparum. So ladies and gentlemen, the smallest eukarya that is found, that is in this option, is option C. So Plasmodium falciparum is the answer to this question, Plasmodium. So the next question is trypsinogen. So it means that question 1, the answer is C, is the smallest eukaryotes. Okay. Then now we are moving to question 2. Trypsinogen is activated by A, chymotrypsin, B, enterokinase, C, trypsin, D, HCl. Okay. So these are the digestive enzymes. These are the digestive enzymes. The enzymes, the trypsinogen, rib, trypsinogen. Trypsinogen is, we call it like proenzymes. Proenzymes. When we use proenzymes, it means inactive enzymes. It means inactive enzymes. So as I said, these are the digestive enzymes. So trypsinogen is an inactive enzyme. And when it is produced, it's usually inactive. It's usually inactive. And of course, this trypsinogen is an enzyme that produced in the pancreatic juice, in the pancreatic juice. So it's produced as an inactive enzyme, so it needs to be activated. It needs to be activated. So what is used to activate these enzymes, to active enzymes, is of course enterokinase. Is enterokinase. That is option B. And of course, when this enterokinase act on this trypsinogen, it will now activate it to trypsin, which is the active form of the enzymes. So once it is activated by this enterokinases, some of this trypsin, that is the active form of the trypsinogen, will also go back and activate more of this trypsinogen. It means that there are two proteins, or there are two proteins that are involved in activation of the trypsinogen. The first one is enterokinase. So it's after the enterokinase, then the trypsin will be activated. And then once we have the active trypsin, then it will now go back to be activating more of the trypsinogen. And we call that autocatalytic cleavage. Autocatalytic cleavage. That is an enzyme that activate its own enzymes. So, and of course, this is similar thing that happened in the stomach, in the gastric juice, and the gastric juice and the gastric juice and parietal cells, they produce an enzymes called pepsinogen in the stomach. Pepsinogen. This pepsinogen is an inactive enzyme. So in the stomach, HCl is the one that activates this pepsinogen to pepsin, which is also active form of the pepsinogen. So once pepsin is being activated, then it will also go back and activate more of another pepsinogen. So this is in the stomach. So the answer here is B. And the next thing is lysosomes. Lysosomes are produced by A, hydrolytic enzymes, B, autophagy, C, destruction of cells, D, all, and E, not. So actually, most of these questions, options is like they are trying to give a function of the lysosome, but there is nothing that actually produces the lysosomes here. So the answer is none of the above. This lysosomes is one of the organelles of the cell that are found, and the main functions of lysosomes in a cell is to clear foreign organisms. When there is any bacteria that try to get into your cell and affect your cell, so in this lysosomes, there is a hydrolytic enzymes. And the name of the enzymes is lysozymes. The name of the enzymes is lysozyme. The name of the enzymes is lysozyme. And what this enzyme does is to cleave, all any foreign microorganisms that want to cause harm in your body. So it will now destroy it, it will now eliminate it. It will now destroy it and eliminate it. So that is what actually happened. So these lysozymes, They are usually, oh yeah, they are usually in most of our body fluids. Like we have it in our mouth, we have it in our saliva, we have it in our eyes, we have it in our mucus, we have it in our sperm. So all the functions of these lysozymes is to try as much as possible to, because most of these openings is where most of these microorganisms are getting it. So that is why we have fluid naturally. And in that fluid, we have these enzymes, lysozymes, that usually use to try to protect us from any foreign organism that want to get into our body. So, and of course, lysosomes, these lysosomes is not actually is the main enzyme that involved in the production, and the lysosomes is the organelles where where the lysozymes are found. And these lysozymes are produced from some vesicles from the Golgi body. So Golgi body is the one that produces some vesicles that joins themselves to produce lysosomes. So it means that lysozymes are produced from Golgi body. And here there is no any Golgi body. So it means that the answer is none. So the answer is none of the above. So the next thing that we are going to look at is question 4. Which of the following statement is incorrect? A competitive inhibitor binds to allosteric sites. B. The substrate binds to active sites. Enzymes worked based in low pH. D. Both A and C. E. None. So, ladies and gentlemen, when you said enzymes, they are proteinous in nature. They are biomolecules that usually speed the rate of the reaction. They usually speed the rate of the reaction. And most of these enzymes, like for example, if the reaction is going to take 10 days, so if you use enzyme, it may take just 1 minute. It might take maybe 2 minutes. So it's just trying to increase, the rate of the reaction. It always tries to increase the rate of the reaction. So these enzymes, the structure of the enzymes appear like this. In the enzymes, we have what you call allosteric sites. Allosteric, we have allosteric sites, and we have active sites. So active sites is a space on the enzymes that usually substrates bind. When we say substrate, it's like in a reaction, in normal reaction in chemistry, we know that we have reactants to products. So in biology, reactant are called substrate. So it means that the active site is a region where the substrate or the reactant binds. So it means that option B is correct. And then we also have allosteric sites. Allosteric site is a site where modulators, also molecules, bind in order to increase or decrease the activity of the enzymes, to increase or decrease the function of an enzyme. That is the allosteric site. Is a region or is another side of the enzymes that some molecules bind in order to either increase the action of the enzymes or decrease the action of the enzymes. And we have inhibitors. Inhibitors are molecules, because once we said inhibitors, it means that there are molecules that always decrease the activity of the enzyme, that always decrease the function of the enzymes. And we have different types of the inhibitors. We have competitive inhibitor, we have non-competitive inhibitors, we have mixed competitive inhibitors, and we have uncompetitive inhibitors. So usually when we say competitive inhibitor, it means that there are inhibitors that are competing with the substrate. It means that they are also binding with the enzyme's active site. So both the substrates and the inhibitors, they will be competing for the same active site. So that is why they are called competitive, because the substrates and the inhibitor, that is the reactant and the inhibitor, are fighting for the same active site. So that is why it's called competitive inhibitor. So competitive inhibitor is not binding to allosteric site, it's binding to the active site. So therefore, this option A is not correct. It's not correct. And then option C. Option C said enzymes work based in low pH. This one is also not correct because the enzymes can work in acidic pH, it can work at low pH, it can work at intermediate pH, it can work at very high pH. It depends on the nature of the enzymes. So therefore, enzyme doesn't work based in low pH. It depends on the types of the enzymes, depend on what the enzymes is doing, and it depends on the nature of the enzyme. Some enzymes work at 10 pH, some work at 8 pH, some work at neutral pH, which is 7, some work at 2.0 pH. That is very, very low pH. So that is this. So option C is also not correct. It means that our answer is D. Both A and C is incorrect. Both A and C is incorrect. So option D is the answer to this question. It's the answer to this question. So the next question, which is question 5, says all of the following are characteristics of fungi, or are characteristics of kingdom fungi except A, heterotroph. Yeah, of course, fungi, they are heterotrophs. They doesn't have chlorophyll. They do not have chlorophyll. And remember, any organism that does not have chlorophyll, it means it cannot undergo photosynthesis. So that is why they are heterotroph. They are heterotroph. And then B, sessile. When you say sessile, it means that immotile or non-motile. So generally, fungi are non-motile. And then the C, cell wall. Fungi also produce cell wall. So these are some of the characteristics of fungi. And then D, sexual reproduction. Of course, fungi undergo both sexual and asexual reproduction. So for the sexual reproduction, is because they produce a chemical called pheromone. Pheromone. So this pheromone is the chemical. This pheromone is a chemicals that allow the fungi to undergo sexual reproduction. And of course, for the asexual reproduction, to undergo spore formation. It's usually using spore formation. Remember that for the asexual reproduction, we have different ways of asexual reproduction in slow organisms or in lower organisms. And examples of those types of sexual reproduction, we have spore formation, we have budding, we have binary fission, we have yeah. So these are some of the example: binary fission, spore formation, and others. So that is it. So it undergoes sexual reproduction, and of course, so it means not. So none of the option is not correct. So it means that E is the answer. So all the options are correct. So not. Okay. Which is E. So then 6. Where is the site for photosynthesis? Where is the site for photosynthesis? The site for photosynthesis. So what is photosynthesis? Of course, when you say photosynthesis, it's a process whereby organisms produce their own foods. So in photosynthesis, plants utilize carbon dioxide plus water in the presence of sunlight. And of course, there must be chlorophyll. So once there is this, the plant can now synthesize their food, which is glucose, C6H12 or 6. So the plant will now synthesize their own foods. They will synthesize their own foods. And this is called photosynthesis. So where this photosynthesis take place in a cell? Where this photosynthesis take place in a cell? Photosynthesis is only taking place in a leaves. Of course, in a leaves, we have what you call chloroplast, and in the chloroplast we have what you call chlorophyll, we have chlorophyll, and in the chlorophyll, we have what you call thylakoid membrane. We have thylakoid membrane, and this thylakoid membrane is what combats, or it's what absorbs the different colors of light spectrum to create energy. So it means that the thylakoid membrane specifically is what converts the light energy into chemical energy that is in the process of photosynthesis. So thylakoid membrane, strictly speaking, is the one that carried out the function of photosynthesis. But of course, as I said, in plants there is chloroplast, and in the chloroplast there is chlorophyll. And in the chlorophyll there is a thylakoid membrane, and the thylakoid membrane is what absorbs the solar energy and utilize carbon dioxide and water to generate energy. So therefore, the answer is option A. Then humans are A, producers, B, primary consumers, C, secondary consumers, D, tertiary consumers. So what is producers? Producers generally are organisms that synthesize their own foods. They are the organisms that synthesize their own food by themselves. They synthesize their own food by themselves. While primary consumers, they are the consumers that get their food from the primary producers. They get their food from there, since they cannot synthesize their own. So human being cannot synthesize their own food, so they depend on other organisms. So human is not a producer, and of course it's not a primary consumers. Human being fall under secondary consumers. So C is the answer. So ladies and gentlemen, I'm going to stop from here, and then we'll watch the next video. Okay? Please, if you are not subscribed, please subscribe to my YouTube channel. If you didn't subscribe, please subscribe.